Miriam, thank you so much. And like I said, we have a bunch more questions coming in. This is really exciting to have access to such a source of knowledge here on spotted lanternfly for all of us who are trying to deal with it out in the field or worried about it coming in. So I definitely hope we can have some time at the end to go back to these questions. But right now, I'd like to make sure we get Dr. Julie Urban's presentation going, because she's got some really interesting work going on looking at management and also at biology. So she's going to give an update from Penn State research on spotted lanternfly management and biology. And Dr. Julie Urban's a research associate professor in the Department of Entomology at Penn State University. So Julie, if you want to go ahead and share, Miriam, if you want to stop sharing your screen, and then Julie can go ahead and share her screen, and hopefully.
Okay. Alrighty, here we go. Okay, great. So I'm currently at Penn State, but I started working on spotted lanternfly in 2014 when it first appeared. At that point, I was a scientist at the North Carolina Museum of Natural Sciences, and I just had the opportunity to come to Penn State mid-2016. And so my research primarily had been looking at the genetics of spotted lanternfly and looking at their endosymbionts. But now, as I've moved into Penn State, because Penn State is the land-grant institution and has a close relationship with PDA, or Pennsylvania Department of Agriculture, we've really there's really been a need for Penn State and many folks within Penn State to step up to provide management options, short and long term, on lanternfly with state funding from Penn State and from PDA. And so what I want to do in this particular talk is I've given a webinar before for New York Northeast IPM, and we have a lot of webinars where I talk about research available online. And what I'm really focusing on today is more of the new stuff that's trying to respond to what the current situation is because this pest, the situation changes from year to year. And if we look at, there we go. If we look at the map from Northeast IPM, I think this was updated, June 11. So slightly, it looks very similar to what Miriam presented. You all are very fast at keeping this updated. But you can see that we have 2 established populations of spotted lanternfly, and more detections are popping up. And the thing, and granted, maybe because I'm in the thick of it and I'm talking with growers and whatnot, but I really feel that this is a dire situation. So take what I say maybe with a grain of salt in that respect. But essentially what we're seeing is that this is moving into, really Philadelphia presently. And what I predict might happen in the next year is that it's going to move into the major urban centers of the Northeast. And that's certainly really problematic. As Miriam mentioned, we know that in vineyards, spotted lanternfly can be devastating, and that's hard to quantify, but we were able to do so last year. Our economists, Jay Harper and Lynn Keim, with our extension person, Heather Leach, who did weekly surveys of 5 impacted vineyards last year every week, and Michaela Centenary, our viticulturist. What we saw that in 2017, there was a 90% yield loss in a 40-acre vineyard, and that is Beekmans Vineyard. Calvin Beekman reported this in testimony in front of the Pennsylvania House and Senate hearings in 2017. But it wasn't, that kind of loss wasn't unique just to Beekman's property. We saw 100% death of a new planting of Pinot Noir in another vineyard, and we certainly saw yield reduction in Chardonnays in another vineyard. And what we saw at the end of last year was that there are more vineyards where SLF is moving in. And looking at the spray records of these 5 vineyards, the average number of insecticide applications applied for spotted lanternfly increased from an average of 4.2 to 14.0 in 2 years. And that increased cost was an increase of 271%. And the really alarming part here is that it still didn't provide sufficient control. And so the problem here is that, it's really easy to kill spotted lanternfly, but more just move in from the surrounding woodlands. And so it's that inundation from the surrounding areas that's really nailing the vineyards. And so Beekmans was really frightening in that we saw last year none of his, like many of his vines didn't survive the winter. Last year, the surviving vines didn't set fruit, and we just went there in May, and essentially it's all dead. And so 40 acres, that's a picture from Beekmans Orchard. It's a really dire situation. And you know, we spoke with another vineyard that caters weddings and different parties using the Cabernet from Beekman and whatnot, and they've run out of it. And there's a good trickle down effect in terms of how the grape industry, how the vineyard industry is being hit in southeastern Pennsylvania. And so that was very, very sobering to see Beekmans look like that. And so as Miriam worked on traps, and so we did another study looking at traps in Pennsylvania and Virginia. So looking at Pennsylvania as an area of high infestation versus Virginia, you know, the established population has a lower density there. And so we used slightly different traps than Miriam did. We didn't have as great a luck as she did, so we're kind of working together this year to improve things. But we used a circle trap, a modified curculio trap, sticky band, and a yellow sticky band, and baited them. And we saw that the methyl salicylate lure really didn't have any effect in Pennsylvania, nor did it really have an effect in Virginia. So this is why Miriam's work is so important, and we're going to work with her this year. And what we're doing, this is a county map. You can see we're going to do this trapping study, but also do weekly monitoring in additional vineyards in 2019. So you can see here we have a series of vineyards that we're working with and that Heather has already set traps at, and we're monitoring weekly. We're also monitoring a nursery, and we can talk about this in a bit, but basically nurseries we're seeing they're particularly hard hit because they're not able to sell spotted lanternfly or sell any plants that have any spotted lanternfly living or dead. And so when you're a 95-acre nursery like Eaton Farms, right up against a stand of Ailanthus that's laden with egg cases, you know, it's a serious cost on their production to keep all of their plants clean. And so we're working with them because they're just being hammered. We're also monitoring orchards, and Greg Krawczyk, a faculty member at Penn State, is really focusing on the orchards, where we're doing trapping studies there as well. So we really want to monitor and see how this damage, hopefully this damage isn't going to extend, but again, I'm not very optimistic at this point. Okay, so what do? We've been doing insecticide trials. Last year with funding from PDA to Penn State, we did a series of trials on potted peach and grape out at the Penn State Berks campus, because our main campus at Penn State is still 3 hours away from the quarantine zone. And so this is what I've talked about in other presentations, so I'm not going to go over that again here. We have a scientific publication that recently came out from that. You're free to consult that. And also we have updated recommendations for tree fruit and grape production on the Penn State Extension website. Also on the website, we have results of trials we did for homeowners last year, and we have recommendations for SLF management for homeowners. And then more recently, again, with a lot of empathy toward Eaton Farms and how this is hitting the nursery industries, there's also recommendations for spotted lanternfly management for landscape professionals. And this year, this is something that perhaps Eric might talk about a bit. We're really looking to expand those and look at Bavaria-based compounds. We know Bavaria is an entomopathogenic fungus, and there's many compounds that use it for control. And so we're really focusing on that. We also have a field trial going. Actually, again, hopefully Eric will talk about that, where Bavaria compounds are being broadcast, and we're looking at the effectiveness there. We're also performing additional studies to refine recommendations for grape and tree fruit a bit. And here, this is David Bittinger, who's one of our fruit researchers, and Heather Leach. Because of the situation that's so dire for nurseries, we're looking specifically at treatments they would use. And again, these are on potted plants at our Berks campus. And we're also looking at trying to target symbionts, the endosymbionts of spotted lanternfly, which I'll talk about in a bit. And then the big thing with lanternfly moving into these major urban areas, and with it being such a nuisance pest for homeowners, there's a real important need to do research on ornamentals. And here, nobody has an ornamental experimental plot. And so this requires finding trees of a similar species. It's often red maple that have been planted at the same time, same age, same size. You need to find kind of great conditions to be able to test these insecticides, because Phil Lewis is doing some really great work looking at pesticide applications on Tree of Heaven, but applications and how, really differ by tree species and whatnot. And so I'm very encouraged this year because Penn State Extension hired an extension educator specifically to conduct this research. So we recently hired Brian Walsh, who has an applicator's license. He has over, I think, 180 trees. It might be pushing for 210 trees in various neighborhoods where he can conduct these important trials. And I think that's going to be super important for giving homeowners and other in the ornamentals industries practical ways to control this thing. And so, kind of summarizing or stepping back here, we know that we see this late season movement of spotted lanternfly into vineyards where they come in at high numbers, and we know that can be devastating. And in 2017, we first saw that moving into apple and other fruit orchards as well. We saw that again in 2018. So you can see here, they're swarming this apple tree. This is what kind of freaked everybody out. And in 2017, this is when spotted lanternfly kind of hit the press, so to speak. But we saw that they really only fed there for 2 or 3 weeks. They did it again last year, and they really didn't do any damage to apple or tree fruit. And still no damage has yet been detected. But we also see this movement onto maples and other trees, and potentially here this is more damaging. And so if we want to think about control, as Miriam mentioned, USDA and PDA have focused on treating spotted lanternfly on Tree of Heaven, which I think is a very important strategy. But the thing is, they're limited. They have limited resources. And we know with the spread we're seeing of spotted lanternfly, we need to hit it at other life stages. And so we have work, and this is in our publications, looking at ovicides. Additional work is being done there because we haven't found much other than Lorsban that's been effective. We know that the early Instars feed on a really broad range of herbaceous plants, and those can be treated with contact sprays pretty easily. Fourth Instars we see, as Miriam indicated, movement onto black walnut, and so potentially that's another potential target. But the thing that's really been, that I've been focused on and that we've really hit on is this late season movement. And this late season movement is something that I think we need to better predict when and where it happens, and we need to better predict what impact it has on these plants. And this is a key additional place, that Penn State can provide recommendations to control lanternfly on things in addition to Tree of Heaven, because we're going to need to hit this on all life stages in order to knock populations down. And so Miriam did a great job in, talking about this development in females. And so we know, the adults are very long lived. They appear in late July. They lay eggs October, November. They usually don't die until December. And if you look at this photo, you can see the red valvulae at the apex of the abdomen there. That shows these are both females. The legs have been ripped off so you can see the abdomens better. But certainly there's individual variation, but here you can see this extreme size difference in these two females, and they were only collected 1 month apart. And so that doesn't even encapsulate the full size difference you're going to see from an early season female in July to late November. And so at that time, they're feeding voraciously. Miriam and I have the same picture here. And so you can see they're just feeding heavily on this particular tree. If you look on the right, this is a homeowner where the lanternfly were flying and flew against the house and fell into the chimney. And so that's the base of their that's just covered with spotted lanternfly beneath their fireplace. The numbers that we see on silver maple, for example, there was a study that we did with our landscaper, Brian Walsh, where a single silver maple that was an attractive host, similar to the tree in this picture, was injected with dinotefuran, so a neonicotinoid. That's what PDA and USDA are using to treat trees. It was injected, and the number of dead lanternfly were counted. And over the course of a week, over 12,000 dead lanternfly accumulated beneath that one tree. And so literally you're seeing thousands impacting a tree, and we don't really know what the long-term effects are on tree health. Certainly we're seeing some immediate death in branches and some flagging, particularly on black walnut, but what does this do sustained over time? That's what we don't know yet, and that's what we need to study. And so what's interesting is that last year was the first year we were really able to quantify this late season movement. But kind of the best place you really see the biology reflected is in the number of calls coming into the call center at Penn State. So with funding from USDA, Penn State has gotten money to do outreach, and part of this outreach involved hiring a group of people to sit in a room and answering questions on spotted lanternfly. And you can see that the spike of those calls coming in per week occurs in that late season movement, and you can even see number of hits to the website nearing 60,000 at that time of flight. And in a particular study where we quantified this as well, this was last year a study that was done in collaboration with Rainbow Tree Care. And this is one of those in situ ornamental studies I was telling you about. We happened to find a series of 40 red maple that were planted on the way up to an adult care facility. And you can see 20 on each side of the street. They were treated with varying compounds in this insecticide trial. And we recorded the number of spotted lanternfly on these trees over the course of several weeks. And what we saw was you got a bit of a bump of lanternfly coming into these trees at the end of August, but then a much larger bump in October. Those numbers you can see averaging between fewer than 10 to 30 per tree aren't alarming. A different study we did where all of the trees were treated with dinotefuran at a different site shows a different story. And so Mason here is Charlie Mason. He's a postdoc who we've recruited to help out with this. And what you're seeing here is each line is an individual tree. And because these trees were injected with dinotefuran, we just recorded dead bodies of lanternfly beneath the tree twice a week. And you can see that top line, that particular tree, we found over 500 dead lanternfly on August 29th, picked them up. 2 days later, there were almost 1,500 more. The next week, 1,000 more. The next week, 1,800 more and whatnot. And so if you think, you think of that cumulative damage on a tree, that's really alarming. And that dip, if you wonder what that is, that was a rainy period. And so we see they don't move so much when it rains. But again, this is the first time we were able to really quantify that late season movement. And this is what we need to focus on. And what does it do to trees? This is a different site where we saw just red maple slackered with egg cases. But based on these observations, this is what we're using to inform the research we're doing this year. And so last year, we looked at some phytohormones, so we know that that level of feeding is going to cause changes in plant chemistry. And so with Charlie Mason and an assistant professor, Semperna Satar. They're both plant people. And so we've enlisted them to work to more fully characterize the phytohormone and metabolic changes in plants. And so what we saw last year, we did an experiment where we exposed silver maple to spotted lanternfly feeding versus not. And we saw differences in cytokinin levels, but we're working on expanding our sample size and looking at those responses in ornamentals this year, again, led by Charlie Mason. But also we need to do the same thing in grapes. And so on the left is a picture last year from Beekmans Orchard, and that's what we saw, the vines that he had surviving at that time. Those that set fruit, it was really puny. And so we're working to really look at what's going on in the physiology of these plants as lanternfly is feeding on it. We had a series of planted grapes last year in a vineyard that we built cages around, where we had started some preliminary work, and that preliminary work where we enclosed different levels of lanternfly, different numbers of lanternfly to represent different levels of feeding damage. And we looked at grape physiology, again, led by Michaela Centenari, a viticulturist. What we saw was it looked that there was a greater that greater feeding was associated with less cold tolerance, but again, very preliminary. Unfortunately, those vines died due to winter damage, and so now we've had to find another place to do this study, and we have Martin Kubek, who has 4 acres of vines, who he agreed to let us enclose lanternfly on 4 rows of his vines and essentially destroy them, but he's a hobbyist, so he's willing to help us out. And so what you're seeing in the picture on the right is, besides looking at plant chemistry changes, we also, that person in the center there is Roman Zweifel, and he's an expert who's from Sweden, and he's developed dendrometers that allow you to measure changes in xylem and phloem movement, both in plants. And so we had a workshop where we brought him and some other folks out, and this is him. You can't really tell, but he's putting the dendrometer, showing us how to put these dendrometers on the grapevines. And that's it all set up. You can see you have to put the cords in some bags, but basically I just got an email yesterday that Michaela has these all set up on Martin's vines, ready to go. And in a couple of weeks, when we have adult lanternfly, we're going to enclose them at various numbers on the plants and use the dendrometers as part of our measurement. Ultimately, we also want to do RNA extractions and look at changes in gene expression. Right now, what we're doing is we're enclosing lanternfly with clip cages on grapevines in the lab to look at that more locally, but we want to extend that to this study at Kubek's as well. Also, the other thing that I'm really excited about is that I bought an electropenetration graph. And so we have Semperna Satar, the assistant professor I was mentioning. She uses EPG to study feeding behavior of aphids, and we bought a bigger system, an ACDC system, where we can do this on spotted lanternfly. And so essentially what you're doing, you can see we have a tree of heaven in a Faraday cage, and we have wires all hooked up, and basically by running, if I show another picture here, you see a little lanternfly in the bottom center hooked up onto a gold wire, and because we have an electrode in the plant, when the lanternfly feeds, that closes a circuit. And so you can read these waveforms, and in it, what the waveforms will tell you is exactly how the insect is feeding. So you can tell when it's probing, when it's inserting its mouth parts into xylem versus phloem, some really interesting things. And so what we see here is very, very preliminary. But basically you see a difference in waveforms between a first nymphal instar versus a second nymphal instar. And what this suggests, again, we have to validate this with some histological studies, but the waveform shown by this first instar nymph is characteristic of like a punching feeding, that it's just blowing through any cells, versus the second waveform actually is typical of a salivary sheath feeder. And so when it seems that second instars when they're inserting their mouth parts, they're introducing a salivary sheath, and actually that tends to be more damaging to the plants that it feeds upon, because it kind of hides the feeding from the plant defensive responses. And this is new to me. We're just getting this equipment going, but by looking at exactly how spotted lanternfly is feeding and penetrating at the cellular level, and ultimately coupling this with the plant responses in chemistry, metabolomics, gene expression, and fluid dynamics, we're hoping that we can really get a better sense of how spotted lanternfly is impacting plants. And so I'm really excited about that work. Other work that's pretty new, I'll just kind of cruise through this, is that the genome of the spotted lanternfly has recently been sequenced. Currently, we're working on the Symbiont, so I'll be leading that part. Eric is going to talk about fungal enemies of spotted lanternfly, and Miriam mentioned Julie Gould and Kim Holmer, who are looking at parasitoids and other natural enemies of lanternfly. And so certainly biological control and classical biological control are wonderful options. I hope they work. It's very exciting. But I think, like with gypsy moth, we're not going to have a single silver bullet. It's going to take a lot of things. And because lanternfly is feeding on so many different species of plants in so many different habitats, I'm concerned that enemies that are already here, if you use those, there might not be the specificity. You might have non-target effects that are really problematic. And so I think that there's room for a synthetic approach, RNA interference perhaps targeting the Symbiont. So work on doing this genome sequencing, I think, is going to be really important to provide perhaps that level of specificity that's needed to help target lanternfly alone. Along those lines, I'm looking at the Symbiont development. So essentially within Miriam's scheme, I'm really focusing on that mid part in detail, because we know that spotted lanternfly have 3 different bacterial symbionts. These are things not just in their gut, they're actually contained in organs. And there are 2 that are known, 2 different species of symbionts, each in their own organs, that are known to occur in Fulgoridae as well as other plant hopper species. Those you can't see here, but there's a 3rd one that's unique to Fulgoridae, not just spotted lanternfly, but I'm currently describing it. And if you can see here, this little inverted sausage, that's actually the structure of this new symbiont. And we have the genome sequence. We're working on annotating them, and that'll be coming out hopefully soon. But what we know is that these symbionts need to be transmitted to the eggs. And so if you look at an early season female and then this late season female over here on the right, the morphology, the position and shape of these organs that are housing the symbionts transforms dramatically, because the bacteria need to proliferate and they need to get transferred into the egg stage, into the egg itself. And so that essential reorganization of the female abdomen is something that I'm looking at in detail, because I feel like if we know what tissues to target and what genes are involved, that could be a really highly specific way to target lanternfly and not anything else. And so again, I'm looking at right now characterizing the endosymbiont transmission, its timing and its variation, and figure out if there's sensitive periods in female development. And we're also testing other ways to interrupt that, potentially seeing if antibiotics could be helpful with Sindhu Krishnakutty at the APHIS lab. And just to mention a few other things really quickly, one thing that really hasn't been done to date is to estimate spotted lanternfly density. And that's really hard to estimate the populations because the distribution is so patchy. And so Dennis Calvin is leading that up on our end. And part of it is, we're looking at doing egg counts and looking at clustering density of these egg counts to try to predict what your sample size even needs to be to come up with a good model. And so that work is currently underway. And we're also collaborating with Matt Helmus and Jocelyn Bem at Temple. And again, Dennis is leading that. So we have some very preliminary data there. Also, Kelly Hoover is doing some dispersal studies in natural forests. And so these are areas where there isn't any Ailanthus. And so she's interested to see how far spotted lanternfly can penetrate. And Miriam has helped design or helped advise this study. I've learned from Sarah Wurzberger based on her work. And so what you see on the left is a spotted lanternfly that's been powdered with a fluorescent powder. Kelly's group will release 2,000 of a particular Instar at a time. So you see on the right, they're all powdered up and ready to go. They're dumped out in the field. And then over the course of a series of 2 weeks, I believe, folks go out at night with their UV flashlights and record how many nymphs they can see. I did this a couple of weeks ago. I did not see a beautiful nymph like that, but you can see they show up really well. On the right, that's a lanternfly nymph. And so that's going to be really interesting to see how far the different instars can disperse in natural forests that don't contain Ailanthus. Kelly is also looking at, do they require Ailanthus? As Miriam indicated, these things are voracious feeders. It's horrible to try to keep them going. Kelly collected, last year, just kind of cruised through this, collected a bunch of 4th instar nymphs and essentially put them in our quarantine greenhouse in one cage. We had 2 cages. Both cages had a lot of trees. One had Ailanthus in it and the other did not. And what she saw was that they only successfully mated and laid eggs in the cage with Ailanthus versus not. But none of those really hatched. And so what Kelly did is last year they built a bunch of enclosures where you have within each enclosure mixed species of trees, some with Ailanthus, some with not. This year, they put the egg cases in it. They've introduced, I think, 200 or 300 nymphs per cage to try to see if they need Ailanthus to feed. And so far, there's very few nymphs that have survived. So to answer the Ailanthus question is really a tough one. Kelly's giving it a go. And we'll see, hopefully she'll have better success this year than it looks like right now, but we'll see what happens. And with that, that's a brief summary of some of the stuff we're up to.
Wow, thank you so much, Julie. That's really interesting and very, very important information to have. And so I just wanted to note, and I don't know if I mentioned this at the beginning, this is being recorded. And so I know there's a lot of information that was in this, both these presentations so far and that Eric's going to present as well, and also some of the questions. They will be recorded and available on our website following the webinar. And so I'm just going to do one question right now so we make sure we have time for Eric's talk in the main time that we had allotted. And then if there's time to go through some of the questions that we already have here, I'll go ahead and do that at the end. And if you have to get off, just know that we'll record the questions as well, so you can hear your answers afterwards if you need to get off. So just one question from Colleen Michaels. There is conflicting information on the residual effectiveness of zeta cypermethrin between the landscape professionals fact sheet and the management for homeowners fact sheet. Can you clarify what the residual efficacy is?
Let me peek right here because I have a previous set of slides about that. But here I have that's the Mustang Max. That there it had good residual through 7 days, but not to 14 days is what I have from the raw data. So I'm not sure. I will follow up with Heather Leach and make sure that we correct that. Thank you for bringing that to my attention. We'll fix that in those publications.
Thanks so much, Julie. And like I said, I'm gonna transition now into Eric, Dr. Eric Clifton's talk. As Julie alluded to in her talk, there are some fungal pathogens infecting spotted lanternflies that Eric's going to talk about. These are natural biocontrols that are looking promising, and I'm really excited to have Eric share this. Eric's from the Department of Entomology here at Cornell University and is a postdoc in Dr. Ann Hayek's lab. And so with that, I will pass the mic to Eric, and I look forward to hearing all about this. So.
Yeah. All right, great. Yeah, so thanks for the introduction, and thank you guys for tuning in today. Just to outline the talk, I will explain a little bit about how we got involved with spotted lanternfly, talking about this Bavaria bassiana fungus, also Batco major, another one we found that caused epizootics. And then I'll talk a little bit about stuff that's going on right now and what we're trying to do in the future here with these bioinsecticides that contain the fungi, and a nice field study we're doing down in Norristown. So this all started back in fall 2017. We had this email going around sent to Anne, and there was a picture of a dead lanternfly adult with this white fungus on it. And we kind of knew what it was, but we asked that they send the sample. And this was found, I believe, in this park of this reservoir just west of Boyertown, and actually not far from, I guess, where lanternflies were first discovered in Berks County. So that was brought back to the lab, and it didn't look like there was much on these cadavers when they came in, but we basically put them on the selective media plates, and this fungus was growing from within the insect. So this was not some kind of saprophyte or mold. It was a fungus that had killed the insect and was inside of it. And we got this on a plate, and with molecular tools confirmed, in fact, that it was this fungus called Bavaria bassiana. If you're not familiar with these fungi, fungal pathogens or insect pathogenic fungi kill insects by spores attaching to the cuticle of the insect, where then they form this aprosorium. They penetrate through the cuticle and enter the body, and whereby these blastospores spread inside the insect, and eventually after a couple of days or more will kill the insect. And then after mortality, then you get the fungus growing out of the body and releasing more spores into the environment. So you can kind of see this is a microscope slide of various spores stained with this blue stain. And you can see here examples of other insects. This is actually Asian longhorn beetle on the right, and a cicada with this Bavaria fungus growing out of it. And currently there are actually multiple biopesticide products that use Bavaria bassiana as an active ingredient. One example here, Botanigard. Another one I'll talk about, Bottega, basically the exact same product using this strain called GHA. So then, in the interest of learning more about lanternflies and about these fungal pathogens, we started to make these field trips down to Berks County in spring of 2018. And we went to this private residence in Fleetwood, which is just kind of northwest of Boyertown, where we got that first sample. So there we were looking at nymphs, looking at egg masses, and me being somebody who hunts for morel mushrooms in his free time, I was digging around the ground, and sure enough, we found these adults that clearly died in fall of 2017, but they were just sort of tucked away under leaves or at the bases of trees, and they still had this white Bavaria fungus growing on it. So brought that back to the lab to culture it, do molecular tools to identify it, and sure enough, again, more Bavaria bassiana seems to be killing these lanternflies. So then fast forward, summer's passing, we make repeated trips, learning more about lanternflies, we attempt to rear them in our lab, and then close to end of September, early October, we got reports from Heather Leach that there were a lot of these lanternflies dying, and there was even another fungus, which I'll talk about later, killing them. And this photo collage is from at least 8 different locations all throughout Berks County. And I'd go to some of these places, and in a given area, you could see dozens of these dead lanternflies on the ground covered with this white fungus. In some cases, they're still actually glued to the trees there. So pretty quickly, we started to get lots and lots of these Bavaria cultures from dead lanternflies. And this little map here shows you that we had at least 130 isolates alone in 2018. We could have collected more, but we basically ran out of cups and space to keep all these. But in one particular location down here near Reading, this is Angora Fruit Farm. There was lots of death caused by these fungi. And this is kind of what it looks like when it goes back to the lab. We create a library of isolates. And when I say isolate, that is one fungus culture from one individual dead lanternfly. And we already started to notice too that they even looked a little different from one another. So you get some that look kind of fuzzy, others that are more powdery. And they all are in this genus Bavaria, but there are different species out there. So we have to do molecular tools to identify them. Even if you look under a microscope, they all have these round spherical spores. So you can't really just tell the difference based on morphology. So we're using a primer pair to identify them at the molecular level. And it's important to do this because we want to know what diversity of Bavaria are out there killing them. Are there some isolates that are more widely occurring or more prevalent than others? And today I can say that we've already identified about 90 of these isolates, and all of them are Bavaria bassiana, but there are still these other species that are known to occur in North America. So that work is ongoing. And then you get just sort of this cloud or this dust of spores all everywhere. So if you imagine lots of insects or a large population in an area, those spores would easily pass to other insects in the environment. So you get this really dramatic sort of tide of spores moving through the environment and passing from one insect to the other. And it's kind of what we think was happening here with lanternflies at this Angora fruit farm site. A lot of them were killed by this fungus that we later would identify as Batco major. Thanks to Kelly Merman, we have this photo here. She called it a bearded lady, and we like that term. We're kind of sticking with it. Right when the adult dies, you get these sort of like white fibers that attach the body of the dead insect or the dead lanternfly to the tree. There's another picture there on the right. And then these wings seem to open up, and then they shoot off the spores into the environment. And so like I said, the wings are extended. One telltale sign that there might be these lanternflies killed by Batco is if you see just like the red hind wings appearing. You can kind of see that here. In the right image, this is at the time we were sampling this epizootic. So these are kind of maybe a quick reference for Batco infections. And at some point, the wings do fall off. And then even in December, when we went back, you could still find some of these dried up cadavers still attached to the trees. So those rhizoids do a pretty good job of holding those bodies to the trees. Some data here, this is showing the sort of the distribution of both Batcoa and Bavaria killing lanternflies at Angora Fruit Farm. And then there's another place just called the Albertus property. That's actually just east of Angora Fruit Farm. And we saw a similar pattern here where in the red is Batcoa, blue is Bavaria bassiana, where if you sampled dead lanternflies on the ground, you had sort of a 50-50 mix of Bavaria or Batcoa. But then if you look at the ones sampled from trees, almost all of them are Batcoa. And that makes sense given those rhizoids that help hold them to the trees. But if you want to learn more about this and the findings from it, we published this paper in PNAS, and it's open access so that anybody can read it. And another point to make there is that we counted egg masses in the epizootic site. We could only find 12 of them. And when we went back in May, we didn't even really find a lot of nymphs crawling around. So it seemed to do a really great job of knocking out that population before peak oviposition. So now transitioning to some stuff we're doing now and looking ahead now that we know these fungi can kill lanternflies. We're going to be testing these bioinsecticides that contain, one containing Bavaria bassiana. In 2018, we did some preliminary trials with Botanigard, but it's basically the same thing as a product made by Certis called Bottega, and they both have a strain of Bavaria bassiana called GHA. But we're also testing a Certis product called PFR97, and this actually contains a whole different fungus called Eiseria fumosa rosea, the Popka strain 97. In 2018, we did some trials where we sprayed tree of heaven cuttings and put the lanternflies on those plants. We did some trials with 4 Thinstars. You can see here on the bottom left, this is a 4 Thinstar nymph with Bavaria growing out of it after exposure to Botanigard. And then down here is an adult with the Bavaria growing out of it. And you do get mortality after about 3 to 4 days, and they had similar efficacy between both products. But I would caution that they weren't the happiest insects when we did these bioassays, because as we learned that tree cuttings are not very good for host plants, and you couldn't even keep these adults alive for a while, partly due to the fact that they're also voracious feeders. But at the very least, this was a good first test to see if these products could actually kill lanternflies. And so We've improved upon that for this year. I have experiments going on as we speak, but we have potted grapevines in our quarantine, and the nymphs do seem to love them. And, we get nymphs molting, showing that they're getting the food that they need. And instead of Botanigar this year, we're using Bottega, but again, it's that same strain, GHA. And we're using what's called an airbrush to spray nymphs directly. This is just a cartoon image on the bottom, but this is the airbrush gun on the right. And so we put nymphs in the refrigerator for about 10 minutes to sort of knock them out. So then they're put on a dish, on an ice pack, and they're cold anesthetized so that they can stay in place and get sprayed with these droplets of these bioinsecticides. And after about like 4 minutes at room temperature, they wake up and crawl around and happy, but they do eventually die. These are just some images of second instars killed by these different fungi. So here on the left is the Bottega, 1 day, 2 days, and 3 days after death. And you can see that because the fungus has infected and killed the insect, it has to grow from within, and it starts to sort of push out between the segments of their legs and body. So you know that it's worked and it's not just some kind of contaminant. And then this is just an image on the right of that PFR 97 product about a week after it's dead. So they also looks kind of white and fuzzy, similar spores growing on it. And I should mention too that this takes about 4 days or more to kill the insects, but we're still collecting the data, so I don't really have like LC or LT50s or that kind of data just yet. And then knowing that these work, we want to carry this out in the field to see if this will be a viable option for control outdoors. So with the help of a lot of people, which I'll name later, the acknowledgements or just show them, we are working in a park called Norristown Farm Park. Norristown is close to Philadelphia, and we marked 4 different areas with lots of tree of heaven, oriental bittersweet, wild grapes, places that have tons of, right now, these 4th instars. And so we established some plots of controls that we get sprayed with water, and then those plots to get sprayed with the Bottega product that has the Bavaria bassiana. And we do this using a air blast gun attached to the back of this tractor. There's just some photos here of John Ross helping to spray the Bottega. And so that's that in action, and you use this air blast gun so that you can really get up there into the canopy and get those nymphs that are sort of out of reach. And we spray till wet, and we're measuring efficacy in a number of different ways. We did timed counts before we did the sprays, and then we're going to do timed counts of lanternflies again 2 weeks after the spray. And then at different time points, we're sampling the population. So we have this nice called a pooter gun or aspirator gun to collect live nymphs from these plots. We bring them back into the quarantine. So here on the far right is some potted grapes. And again, we check these pots daily to see if there's dead ones, and then we throw them on the agar plates to confirm if the fungus is infecting them. But this was just sprayed on July 2nd. We've just started to get some nymphs dying, so too early to say right now how well it's working. But Nina Jenkins has just told me yesterday that some of the leaf samples they collected and washed do have really good numbers of Bavaria on them. So we're hoping for good results. And another way to collect or measure efficacy is we have these sort of ground cloth tarps set up in different areas in the plots. I think there's 4 of them in each plot. And so if any of the nymphs die and fall out of the canopy, they land on this cloth. And so we scoop those up and also bring them back to the lab to see if the fungus grows out of them. As well, we're testing residual activity, we think, with both products on peaches. Julie mentioned a little bit about those chemical insecticides. So we're sort of doing a similar thing where we will spray these bioinsecticides on peaches and infest them with 4th instars in adults. And we should be starting that later this week if the weather holds up. And so David Binninger is involved with that. So lots of stuff going on. Otherwise, there is some other work we're doing with Batco. Knowing that this kills lanternflies, we want to know what else. We'll just expand our knowledge about this fungus and its importance in the ecosystem. And we already know about a few other insects that are killed by Batco, but it's a really understudied group, so we'd like to do more work on that. We know there's Batcoas that kill millipedes. They normally are not found at the top of logs like this, but they've crawled at the top here, and we think because the fungus manipulates behavior and they can release their spores out. And we're sort of doing this in our own backyard, going to some parks close to Cornell campus. Here on the right is a click beetle we found that is attached to some sort of plant stem, and it has that similar sort of behavior or mortality where the insect has died, attached to some surface, and its wings are opening. So Anne's focused on that, but we're excited to learn more about Batco. So to summarize, we know about at least 2 different fungal pathogens that are naturally infecting lanternflies. This is Bavaria bassiana and Batco major. We saw this epizootic, or you could say an epidemic, that killed most of the population there, and this whole woodlot was just inundated with spores to wipe out the population. We know now that Bottega and PFR97 can kill second instars, and we just started bioassays with third instars. And field trials that I just described are currently underway, so results are pending there. And again, looking for more species of Batcoa and improving our understanding about these pathogens killing lanternflies. Here's a ton of people to thank, but I think we'd like to have a little more time for discussion, so we'll get to that when we can. And thanks for listening.
So I'm going to start with our first question here, coming from Charlotte Malmberg here at Cornell. She would like to know if any individuals have demonstrated resistance to the fungal pathogens.
Do lanternflies have resistance to fungal pathogens? We don't really have any evidence of that just yet. I guess we'd have to do more of these spray bioassays or just find populations that don't die from them. But basically, it's too early to tell right now.
Okay. And then we have another question for Eric specifically on how do the fungal bioinsecticides affect non-target insects, especially pollinators? Well,
a high dose of the Bavaria can certainly kill honeybees if you spray them directly, but if you look at the label of the product, it does specifically tell you not to spray near honeybee hives and at certain times of the day when there's pollinator activity. But I'd like to think you treat it the same way you treat other pesticides, where you read the label carefully and you try your best not to affect bees. We don't want to do that. But as far as other non-targets, we have found a few insects at the Angora fruit farm that were killed by Bavaria. But based on the molecular identification I've done, they are different strains of Bavaria and not the same ones that were killing lanternflies. So they might have just been incidental infections because it was a wet time of year, and Bavaria does kill other insects in the environment.
Okay, great. Now I'd like to go back to if Miriam and Julie want to unmute their mics. I've got a few questions that we didn't get to earlier, and we might just take a couple of extra minutes to see how many we can get through. So this question is from Joshua Beast. Do you know of any attempts, if any attempts have been made to use dogs for detection of spotted lanternfly? And this could be for anyone that wants to jump in, Miriam or Julie.
I don't know if Miriam, oh yeah, Miriam, you would know this more than I would, I think.
Well, I think it's a very good idea. I don't know that it has happened at all, but they've been using dogs for ALB and other things, so I think that it's possible that that's something they might try. But it takes a long time to train the dogs to an odor, so I don't think they've done it yet. I don't know if it's in the works.
Yes, it is in the works. Greg Para, who's the USDA person who's in charge of the Spotted Lanternfly Technical Working Group, this is something that he's been involved with. I don't think it's very far along, but I know that it's something that at least is in the planning stages.
Great, thank you. Okay, so our next question comes from Jean Pascal. Cold resistance, how far you think they will move north?
Personally, this is Miriam. I think that they could, because of human-assisted movement, they could pop up on the West Coast. There's no limit to where they could be finding them next. It's their movement without human assistance is not going to be as it's going to be more predictable, but that's not the biggest threat.
This is Julie. I'm trying to pull up, if I can share my screen. There's a map from USDA that, can you see that? The unsuitable area for survival of spotted lanternfly?
Yep.
Okay, so this is, so basically, this is in terms of the overwintering survival of the eggs. And so that's showing you in brown the areas that would not be hospitable to lanternfly based on extension of the Korea data here to the United States.
Okay. Well, thank you. Okay, so our next question comes from Caroline. It looked like some of the negative, this is specifically for Miriam. This came in after her talk. It looks like some of the negative results in the development test were sample sizes of 2 or 3 individuals.
Yes, there were a few species that we were not really, we have very, limitations. We can't do N of 10 on 25 species. We just don't have the time, the resources, and the personnel, and the trees. So we had first choices and then second choices of the trees that we were most interested in, and the second choice trees had lower N.
Okay, thank you. Okay, so now our next question comes from Molly Hassett. She would like to know, what do you think of the genetic diversity of SLF on host selection?
Miriam, do you want to take that on host selection? I mean, certainly we see a population bottleneck. We believe it's only been one introduction, but maybe Miriam can speak to that on the host suitability.
So can you repeat the question?
Sure. What do you think of the genetic diversity of SLF on host selection? Maybe what do you think of the impacts of genetic diversity?
Like Julie said, I think that there is a huge bottleneck from the introduction. I don't know how, I think, Julie, isn't this probably from one introduction?
Yes, it's definitely, yes. Every time they pop up in a different area, they sent them to me to test. And so, yeah, we're still at a single introduction. And
yeah, so I don't think we have a whole lot of diversity in our population here. It doesn't seem to have too much of an effect. They're so they have a very generalist diet. Like they're opportunist feeders, and they're feeding as they move around on pretty much everything. They'll probe whatever surface they're on, even plastic. I've seen them probe plastic just to taste the surface that they're on. So it seems to me like they will opportunistically feed on any species that they happen to be on. They're attracted to certain compounds that are, very, well, they're in Ailanthus, so there's definitely odor mediation of their behavior. Like they are, certain hosts that have those compounds are more attractive. One of the things we just discovered is the tulip tree. That's the tree that only one of them survived out of 100 to adulthood. So it's not a suitable host, but it's anecdotally been shown to be very, very attractive, and a lot of them are feeding on it. The compounds that are in tulip tree are in high abundance, and those the same compounds that are the damaged odors from Ailanthus, and damaged Ailanthus is more attractive than non-damaged Ailanthus. So it's, it may be incidental that they're attracted to all these trees that they're not so suited for their diet. But as generalists, they may be just taking a meal here and there for a little energy, but for the nutritional development, they may need things that are only in certain other species.
And I think if I could add to that, we're seeing the last 2 years what seems to be a moving front, in that last year, when when nymphs were hatching out in the core areas, like in Ole and areas that were just inundated the previous years. Individuals, a lot of homeowners reported that, where they had had thousands, on trees in their yards in previous years, hardly having any. And those were areas that were treated, where the Ailanthus were treated, and areas that were untreated. And my hypothesis, and this is consistent with what people in the field, in the Corps are reporting last year and again this year, is that you get this, it seems that you get this buildup of high numbers over the course of a couple of years. And then I think that they've depleted those plants and they move on. And so
Right.
you see this moving front, and where they move on to are previously undepleted trees. And because they're in such high population numbers, they're kind of going, if Ailanthus is there, they'll certainly prefer it. But I mean, they're just going out to what's next. And so I think that especially with that crowding, certainly there could be some genetic basis for host plant selection and preferences and whatnot, but I don't think we're seeing that. I think we're just seeing they need to feed a lot, and they're just moving on that are undepleted.
Yeah, I agree with that. I think that they, their behaviors are very plastic, and they're very adaptable to the different plant makeups of different areas. And so, and I saw someone was asking a question, so would you focus on detection of the early two on Ailanthus? And I would say, well, I mean, it's a tough question because some areas may not have Ailanthus, but they may have other hosts that are suitable. So I would say that those kinds of questions need to be looked at based on where the high risk areas are and what kinds of plants are there.
Great, and that kind of segues into our next question from Tom Colarusso. He's saying that there's no tree of heaven in the Adirondacks in Champlain Valley of New York, and he'd like to know how you think this could affect the introduction of spotted lanternfly into some of the more northern areas in New York.
May I start? I think that if you have a point introduction of spotted lanternfly with maybe one individual laying one egg mass, you're going to have nymphs that disperse to feed. And at some point, they have to find each other again. And it seems to me, and this is not, tested experimentally, but based on what we've observed over the last few years, it seems to me that Ailanthus really helps them find each other. So I think if you don't have Ailanthus, it may help prevent a population from establishing in the first place when you have a very low number of insects introduced. But if you don't have Ailanthus, there are still plants that they can develop onto adulthood. I don't know if those plants are all going to produce viable eggs, but my guess is they probably will. And so, sorry. So I think that, if in the absence of Ailanthus, there's a list of host plants, especially the ones that I showed as producing adults, that I would look for in areas that are high risk of introduction.
And I would add to that, and I think certainly
Cherry also, by the way.
I mean, I think that, especially if there's grape, in those areas and whatnot, that you might not have sustained populations, but they could consistently reinvade. They might not successfully reproduce there, but
Right.
if the population in surrounding areas is high enough, they're just going to keep reinvading. And that's when they're doing that damage, is the late season, is that, feeding so voraciously in that late season period. So it could still do damage even if they're not persistent there.
Yeah. I think that there's a long list of hosts that they really do seem to like, whether it's nutritionally suitable or not. And so those hosts should be included in the possible hosts that one would look for them on.
I found a map of Ailanthus distribution, if I could show that. This is from a Florida plant database, but I know, and so you can see it's just like a county level. And so you can see that it's pretty much, you know, in a lot of places, certainly densest in the Midwest. But I know that mapping Tree of Heaven is something that currently Penn State has gotten its educators involved with. And then Carrie, I don't know, is this something that PRISM is doing as well? I think that isn't there a New York effort? I think mapping the distribution of Ailanthus is something that is currently underway, is my understanding.
Yes, it is actually. There's a campaign right now with IMAP Invasives and the DC to try to get people to go out and map Ailanthus. So if you want more info, if you're here in New York and you want more information on that, reach out to the IMAP Invasives folks and they can direct you how to get involved with that. So I'm going to wrap up here. I'm so very grateful that our three speakers took time during the most busy time of their year, I'm sure, to share this information with us. It's so important that we have this up-to-date scientific information that they're working so hard to generate to help us improve our decision-making and for us as non-researchers to get out there and actually help them with the data collection and support their work. So I'd like to just thank my speakers again for this great series of talks today and for taking their really limited time to be here. And I encourage you all to check out the website, the New York Invasive Species Info website, nys.info, to look at the rest of the events that are scheduled for the week. We have events all across New York State to get involved in Invasive Species Awareness Week. So we encourage you to do that. And thank you so much for participating today and have a great day.
Thank you for having us.
Yeah, thank you, Carrie. Thank you, everyone.
Cheers.